in particular. So I won't dwell too much on what we know about EHE so far, because I'm sure you all are aware. But what we know is that it's a rare vascular sarcoma, and it has a very heterogeneous disease presentation. And there's two fusion proteins which define it, TASCAMT1 and YAPTFE3. And in this project, it's TASCAMT1 which we focus on. And studies have shown that this is thought to be the driving mutation, and is the only mutation in around 45% of cases. However, there is currently a lack of in vitro models using primary endothelial cells. So this is where my project kind of comes in. So yeah, the first aim really was to actually generate this model. And to do this, we were using mouse embryonic stem cells, and then initiating a differentiation program to generate endothelial cells. And these cells contain a doxycycline-inducible system. So this means that we can just add doxycycline into the cell culture, and TASCAMT1 will begin to be expressed. And from this, we can track the cells which have been successfully induced by looking at either flag epitope or by GFP. So that allows us to do various different assays on live and fixed cells. So it makes it really versatile. So this is just a quick overview of the differentiation process itself. So we start off with the mouse embryonic stem cells in normoxia, and then we generate these embryo bodies, which are, it's a 3D culture of like floating balls of cells essentially, and they're a bit of a mixture of various different progenitor types. And from this point onwards, we use low oxygen. And after 5 days, I then sort out this Tie2 FLIC1 positive population, which is shown here by flow cytometry. So this represents an endothelial progenitor population, which I can then sort out that population, replate them, and after around a week or so, you get endothelial cells, which you can keep in culture for a while and do various different assays with. So this is just showing that you can do, they can form tubes in matrigel, and this is just some flow cytometry analysis showing the expression of a few endothelial markers. So these are a few key advantages really of the model. So the main one really is that we're looking at contextually relevant cells, so they're primary endothelial cells, and it's relatively quick to generate these cells. You can have a lot of endothelial cells within 3 weeks, and you can scale the experiments, scale the differentiations up or down depending on how many cells you need for whatever assays it is. Likewise, you can study the effects of TASCAMT1 immediately upon its expression. So these IF images and some flow cytometry on the side here is just 4 hours after I added doxycycline to some endothelial cultures, and you can see some strong nuclear expression of TASCAMT1, and again by flow cytometry here. And also, importantly, when you add TASCAMT1 to these endothelial cells, they do replicate the transcriptomic features of EHE that's already been published. So again, shows enrichment here of the YAPTAZ signature, and also shows enrichment of the EHE gene set that was published last year by the Rubin lab. So from that part of the project, obviously, we managed to successfully generate the endothelial cells, successfully optimize the differentiation program. And then obviously the next step really was to investigate TASCAMT1 within these cells. So as a starting point, we picked to look at cell cycle proliferation, obviously very, very important for cancer, but we knew that wild type TAS had a well-defined role within this. And again, we wanted to look at some effects of TASCAMT1 expression immediately as soon as it's expressed. And again, also looking at any transcriptomic changes, like all of this within endothelial cells. So here we have just like one of the first experiments that I did, whereas I just looked at the expression of TAS-CAMT1 over time after adding DOX. And what this showed was that the TAS-CAMT1 expressing populations decreased as time went on. So you can see in the summary plot below, you start off with around 15 to 20% flag high cells, and this just gradually drops off as time goes on to the point where it's significantly different by the end of the experiment. This kind of suggested to us that there was some kind of growth disadvantage to the TAS-CAMT1 high expressing cells. So we then went to look on at Ki67 expression, which marks proliferating cells. And what we saw was that it seemed as though upon the initial expression of TAS-CAMT1, you get this big increase in Ki67 positive fraction compared to all of the other, towards the TAS-CAMT1 lows and the non-expressing populations. But this decreased dramatically by a few days later. So this is suggesting that there's an initial burst of proliferation upon TAS-CAMT1 expression, but this isn't sustained and is reduced dramatically after just a few days. So next we looked a bit deeper into the cell cycle and looked at the kind of distribution within the cell cycle phases of TAS-CAMT1 expressing endothelial cells. So I did a cell cycle synchronization here to synchronize the cells to the G1 S phase boundary. And then you release the block and then follow the cells as they kind of come out of this block. And what you can see is that the TAS-CAMT1 expressing populations, both the high and the low expressors, not only are they more difficult to block in the first place, they seem to accumulate in this late S phase here. So this is suggesting to us that these aren't actually cycling any faster than the non-expressing populations. They seem to just be going into S phase and stopping there for a bit. So all of this kind of told us that actually TAS-CAMT1 expressing cells don't really have a growth advantage over the non-expressing cells. They aren't proliferating any faster. If anything, they're becoming arrested and proliferating slower. So we then wanted to really kind of explore the mechanism behind this. So for this, we went to look at DNA damage. This is a well-described mechanism behind S phase arrest. The first kind of experiments we did with this was just some imaging experiments looking at gamma H2AX expression. So this is like a really early step in the DNA damage response by the histone 2AX variant gets phosphorylated by the ATM-ATR kinases. And this then initiates a DNA damage response downstream of that. And what we noticed was that the TAS-CAMT1 expressing cells had significantly more gamma H2AX foci than the uninduced controls. And the number of foci that they had was actually very similar to the peroxide positive control, as peroxide is known to cause double strand breaks. And interestingly, this effect was also visible by only 4 hours after adding DOX, so immediately as TAS-CAMT1 begins to be expressed. And also this was present when you express the T binding mutant version of TAS-CAMT1. So this is this S51A mutation, which again showed a very similar distribution, a very similar amount of gamma H2AX foci as the peroxide and the TAS-CAMT1 wild type expressing cells.
A little bit over to your left.
And then again, we just kind of further confirm this by a different assay. So this is a COMET assay here, which basically looks at DNA fragmentation. So any undamaged DNA stays within the COMET head. Damaged DNA will migrate to form the tail. So you can see that uninduced, you've got like perfect circle. In all of the other conditions, you've got a tail. And again, just represented here in the summary plot, when you calculate the tail moment, the TAS-CAMT1 and the TASCAM21S51A expressing endothelial cells have significantly higher tail moment than the uninduced controls. So from this, we then wanted to see what it is about TASCAM21 really that's causing this DNA damage, because we knew that because it's present from around 4 hours after its expression, or 4 hours after adding docs, this must be something that TASCAM21 is doing itself. There isn't really time there for another pathway to become up or down regulated. So for this, we then turned to looking at replication stress and mechanisms of that. So from some RNA-seq experiment that we did, we noticed that there was a very large number of differentially expressed genes after just 24 hours of TASCAM21 expression. So this was particularly in the TASCAM21 high compared to the uninduced endothelial cells. We had around 5,000 differentially expressed genes. So we thought that maybe this could be some kind of hypertranscription that's going on. And this kind of fitted with a few things we'd seen in the just looking at the log-fold change of the expression of RNA polymerase subunits. We could see an increase in expression of those over the uninduced, and also enrichment for RNA metabolism gene sets. So this is suggestive of hypertranscription. And to further confirm this, we looked at, did some more imaging experiments really. So this nascent RNA imaging works similarly to the cell cycle experiments that I showed you earlier, except you add a different, you add a uridine analog for an hour, and that will incorporate into any RNA that's transcribed during that period. And then I can use click chemistry to image that and quantify the amount of nascent RNA that's been transcribed there. So we saw that there was, again, a significant increase in the amount of nascent RNA in TASCAM21 expressing cells. And a similar pattern was shown here as well with the with R loops. So these happen physiologically as a transcriptional intermediate. They're like a hybrid between the DNA and the RNA. However, if you get an increase in them, and they aren't resolved as fast, and they can lead to DNA damage as well, and again are indicative of hypertranscription. And once again, we saw a significant increase in the fluorescence intensity of the R loop signal in the TASCAM21 expressing cells. So the next step for us really was to go a bit deeper into the DNA damage repair, like what mechanisms there are they working properly, that kind of thing, because there was such an overwhelming amount of damage, it seemed, from our initial experiments. We wanted to see whether this was being repaired. And because we'd shown initially that TASCAM21 was causing an S phase arrest, we looked predominantly at homologous recombination pathway, just because this dominates in S phase and is also the most error-free of the pathways that exist. So the first protein within this pathway that we looked at was BRCA1. So this is a key scaffold protein really in the homologous recombination pathway. It acts at multiple points, but one of its main functions is to bring BRCA2 and PAL-B1 to allow RAD51 deposition, which I'll get onto next. And what we found was that in the TASCAM21 expressing cells, there was still BRCA1 foci formation, but it seemed to be reduced compared to the peroxide controls. And importantly, the docs treated and the peroxide controls had similar amounts of DNA damage to begin with, but there was significantly less BRCA1 in the TASCAM21 expressing population. And then again, this is looking at more specifically at RAD51. So this is, as I said, downstream of BRCA1. This deposits on the overhangs surrounding the double strand break and allows invasion into the sister chromatid, which is used as a template for error-free repair. So this showed a similar pattern, albeit a bit more pronounced. So you can see, again, similar amounts of gamma H2AX in the docs and the peroxide controls, but significantly less RAD51 deposition. So this is suggesting to us that you have all of this damage, but the TAS-CAMT1 expressing cells seem to struggle to repair it as well as the non-TAS-CAMT1 expressing counterparts. And then the next aim really was to look at how does the DNA damage accumulation and the impaired homologous recombination kind of contribute to EHE progression and development. So again, from what is already known, we know that unrepaired DNA damage in a lot of cases will lead to cellular senescence. So this is a type of growth arrest, and it's a tumor suppressive mechanism really to try and stop any mutations from being passed on. So these cells, they don't grow, they sit there. They're very resistant to apoptosis, and they have a very distinct morphology and express P16, which is a key regulator of senescence onset. Or the other option really is where, because homologous recombination seems to be impaired, that more error-prone pathway may take over in this situation and cause a secondary mutation to be acquired. So obviously some of these would be completely inconsequential, but some of them might allow bypass of senescence, might allow a bypass of that initial growth arrest that I just spoke about earlier. So we wanted to look really at both of these scenarios. So what we found initially was that there was an upregulation of P16 expression in the TAS-CAMT1 expressing cells. Normally, P16 isn't really expressed in endothelial cells, so you can see here by the relatively low expression in the untreated controls. And interestingly, there would seem to be cells where you would get P16 in the nucleus and TAS-CAMT1 in the cytoplasm, so it's kind of suggesting that there might be some kind of transition between nuclear TAS-CAMT1, which gets moved onto the cytoplasm when P16 becomes to be expressed. And again, we then looked a bit later on and we noticed these cells in the culture that had that typical senescence morphology, so quite broad and flat. And then again looked at senescence associated beta galactosidase staining. So again, just a really well-known way of defining senescent cells. And again, this showed that in the GFP, also I should mention these cells were sorted originally based on their GFP expression, cultured separately, and then the beta gal staining was done. And you can see that the GFP high and the GFP low have significantly more beta gal positive cells than the GFP negative and the DOX negative, which have a very low amount. So together, this is suggesting to us that this growth arrest that we see initially is down to DNA damage, and because it's not really getting repaired, it's just as time goes on, it's leading to senescence, and then these cells just don't really grow very, very well at all. So then the next thing we wanted to look at really was knocking out CDKN2A. So CDKN2A is being found to be the most common secondary mutation in EHE, and these tumors seem to be more aggressive. But what's interesting is that this encodes P16, so this gene is really important for the positive regulation of senescence. So to look a bit further into that, we did a CRISPR-Cas9 mediated knockout of CDKN2A in both TAS-CAMT1 expressing endo and ones that aren't expressing TAS-CAMT1. And one of the first things we noticed was these really big, bright GFP positive like foci within the culture. And you did see these when in the non-knockout TAS-CAMT1 expressing endothelial cells, but they were much smaller and much like just not as obviously this is really, really bright, really obvious. And this was translated into the TAS-CAMT1 expression here done by flow cytometry, this time looking at the flag epitope. We could see that as the time in culture went on, we did see an increase again in the TAS-CAMT1 expression, but this was much more pronounced when we knocked out the CDKN2A. So again, in the summary plot here, you can see that a significant increase between day 7 and 14 for the knockout, but not for the TAS-CAMT1 expressing control. And then the next step really here was to look at proliferation. So just looking at the cell number, we show that after 7 days, the TAS-CAMT1 expressing endothelial cells have significantly less cells within the culture, but they are able to overcome that, but only to the same level as the non-expressing controls. However, when you combine CDKN2A knockout with TAS-CAMT1 expression, you get this really big significant increase in cell number over all of the other conditions. And again, this is translated into Ki67 positive, the Ki67 positive fraction as well, which again increases significantly in the knockout conditions. So we could show there that how the knockout of CDKN2A can overcome senescence. But what we wanted to do now was to look a bit further into the interaction of TAS-CAMT1 and TEAD. So obviously all of the previous publications had shown how important that this interaction is, and that TAS-CAMT1's interaction with TEAD is definitely needed for its tumorigenesis. But obviously our initial data showed that the S51A TEAD binding mutant could still cause DNA damage, so that suggested that that was independent of its interaction with TEAD. But here we wanted to look in the more longer term cultures and see what happened at later time points. So what we can see here is that when you add the dots after a similar length of time in culture, you don't see any foci formation. The cells just remain as a monolayer. And again, you can see here that there is a slight trend up in terms of the expression of the TAS-CAMT1 S51A, but although it's significant, it's at a much smaller extent than the previous experiments with the knockout and with the TAS-CAMT1 wild type. And what we found, interestingly, was when you use the same timeline as the previous experiments that I showed you, you actually, with the TAS-CAMT1 S51A expressing cultures, the number of cells actually starts to decline after around 10 days, and the whole time remains lower than the untreated controls. And again, this is also shown in the Ki67 staining. So this to us is showing that although the DNA damage might occur independently of the interaction with TEAD, this is definitely needed downstream of this, further on in the culture, to be able to maintain proliferation and overcome the growth arrest. So yeah, this is just a little schematic really of kind of overviewing everything that I've just talked about today. So just showing really that we found that when you express TAS-CAMT1, you get this dysregulation of transcription, which leads to replication stress, DNA damage, which leads to cell cycle arrest. And because this damage is struggling to get repaired, in the vast majority of cases, these cells become senescent, which means that they're not dividing. And we reckon that this could maybe represent a more indolent EHE. However, there's always a chance that error-prone repair might take place, and a secondary mutation could allow senescence bypass, and this could represent the switch to a more aggressive EHE. So this is kind of what, in the future, what we reckon we could do with this model, with this information that we found out so far. So obviously the big one would be drug testing. So finding out that these cells are impaired in homologous recombination would suggest that PARP inhibitors could be useful, and that's something that I'm optimizing at the moment to look at viability after PARP inhibition in these cells. But also various other inhibitors that could be useful. So like CDK4 and 6 inhibitors could replace the activity of CDKN2A if that's been deleted. Again, targeting the TAS-CAMT1 TEAD interaction we've shown might dampen the ability of these cells to proliferate in longer term cultures. And again, ATM-ATR inhibitors could act in a synthetic lethal way to cause apoptosis instead of senescence in these cells. And again, you could test combinations of these as well. Also, it would be interesting to find out how TAS-CAMTA1 interferes with the homologous recombination pathway. Again, this could be something to do with the protein itself causing, like, getting in the way of the homologous recombination proteins, could be binding to them. We're unsure about that, but if we did find out, then this could, again, provide insights into potentially useful therapies. And again, because we've shown that the CRISPR-Cas9 knockout is successful in these cells and is relatively easy to do, we could then use this model to investigate the effects of other secondary mutations that are maybe found in other patients, which could then allow more personalized testing of medicines. So yeah, that's all that I'm going to talk about now anyway. This is just the acknowledgements slide. So yeah, any questions?
So I have a question. Can I jump in? Or?
Yeah, of course.
So this is Ajay Pabat. So you showed really nicely about how the TAS-CAMTA1 is upping the DNA damage. I mean, how much of this is clinically relevant, or how much of it is an artifact of your system? I mean, do you see like H2AX staining clinically in EHE samples, and you see like a correlation between that and TAS-CAMTA1 expression?
Yeah, so we haven't actually done anything in patient samples, but that would definitely be interesting to look at. I think the thing would be is that it seems quite likely that these cells could be becoming senescent, and DNA damage would definitely explain that. And I think because the cells becoming senescent, that explains as to why EHE isn't particularly, it doesn't have a huge mutational burden, because the cells become senescent before any mutations can be passed on. But
the tumors also grow really big. I mean, at some point, you should have this proliferation. I mean, like you show it in your in vitro system, you know, there is an initial proliferation phase, and then it kind of tanks, but it's in a few days. But in a tumor system, you know, somehow it allows proliferation, tumors to be bigger, and then it probably
I guess it's because it could take either pathway, really. Like, in some cells there'll probably be so much damage that they just become senescent anyway, but in some cells they might try and repair it, and obviously some of those mutations might be completely inconsequential. But I think that the paper that I showed that looked at the secondary variants did show that there was obviously, the more mutation, like secondary mutations, the more aggressive the tumor would be. And I think that really fits with what we've shown, because we showed that as soon as you get at least one secondary mutation, you're really increasing the growth. So yeah.
Thanks.
So I guess I'll jump in. Emily, nice job. So I'm still, I think we might have touched upon this in Telluride, I don't remember, but I'm still fascinated by the fact that the S51A is inducing DNA damage. Mm-hmm. So in your model, you think the DNA damage is this massive uptick in transcription, but in theory, that mutant shouldn't be driving transcription, at least not the way we understand it working. So I think I asked, you didn't look at the transcription output in general from the S51A, you just looked at DNA damage, right?
Yeah, we haven't yet. It's ongoing. It's in the pipeline. That's what I'm going to spend the last couple of months of my PhD doing, really, is just kind of repeating some of those experiments with that mutant. Yeah, I do think that obviously if the DNA damage is to a lesser extent in the S51A cells, but is still significant compared to the uninduced. So it could be that this is coming from different, just from different transcription factors compared to TEAD, other than TEADs. But yeah, that's something that I'm working on now, so we'll see.
Okay, so I mean, I guess it would be surprising to me if it had the same transcriptional upregulation. Do you think there's some other way that the DNA damage could be getting caused, and the transcription is kind of a red herring? Or
no, I mean, Oh, go on, Valerie. What was you going to say?
Yeah, just I want to add to what you just say. Is like when we look at the transcriptional landscape upon TAS-CAMT1 addition, only a small fraction of the genes that are dysregulated we can assign to the TAS signature. A lot of the genes that become hyper-transcribed are not related to TAS. So we think that the CAMTA1 is really important. And, there's been publication showing that CAMTA1 interact with the attack complex. So maybe initially you get a lot of change in the transcriptome that are driven by CAMTA1 rather than by the TAS TEAD.
Yeah, I mean, I think that's reasonable, but and Munir's on the call, maybe he can correct me, but the attack and the chromatin landscape does require TEAD binding, at least in their system. But again, who knows what's going on in EHE itself. But yeah, so it would be interesting to see if the transcription is up, but it's a different set of genes. That would be really interesting.
Mm-hmm.
So I guess we'll stay tuned.
Yeah.
Yeah, I agree with John's comment. I mean, even in our model system, it does require some TEAD binding. But yeah, I mean, it does raise the question of whether some of the double-stranded breaks could be due to epigenetic complexes recruited by the fusion protein. So I kind of agree with both comments. I think TEAD binding is important, but you can envision a scenario where the CAMTA1 end is still causing some DNA rest due to recruitment of epigenetic complexes. And the potential explanation in the absence of TEAD binding would be that CAMTA1 does confer some novel DNA binding properties of its own. And so it's possible that in the TCS51A mutant that it's still able to bind DNA at non-TEAD consensus binding sequences. That would be one potential explanation.
Mm-hmm.
I don't know if that came through. Yeah,
sorry, I was just going to say, just as a general comment, I think identifying these places where someone's research throws up an idea that doesn't quite fit with our thinking or model is really interesting, because it may be that the thinking is wrong and the conclusions are wrong, but it may also be that it's highlighting something we still don't understand about EHE. And so I think as part of this process, if people have time afterwards just to document very simply things they think would be quite good for someone like Emily to pursue. I'd like to understand why you're seeing this, or this doesn't quite fit. That would be a great outcome from this sort of meeting, I think.
Mm-hmm.
So Emily, quick question, I might have missed it. Your potential BRCA phenotype in terms of the RAD51 staining, did you see the staining at baseline without any external induced DNA damage?
No, so in the cells that were completely untreated, like just the endothelial cells, you would get a little bit of staining, but it wasn't anywhere near significant compared to the others. I think I can go back and show you. Whereabouts are we? So yeah, the RAD51, you can see here that there are a very low amount of gamma H2AX, but these seem to show again a similar amount of the RAD51. So in these cells, the small amount of cells that are damaged, they seem to be competent in being able to repair the RAD51 by RAD51.
Sorry, and so because the BRCA is basically the loss of RAD51, right?
The loss of BRCA1.
No, but in terms of, let's say, a biomarker, because we talked about the potential of using a biomarker to measure in tissue, and the challenge with using a biomarker like RAD51 is that you need to have appreciable endogenous DNA damage to be able to measure that biomarker. If not, it would need to be artificially induced, which would mean you cannot use formalin fixed tissue, for instance. So I'm just wondering what your thoughts are in terms of.
So I think.
Yeah, you do get the PARP sensitivity, let's say, in terms of thinking of a biomarker driven strategy.
Yeah.
RAD51 would be a good candidate to look at least from the clinical experience with prostate and breast and ovarian.
Yeah, because I think the thing is if you look at purely in terms of the expression of the protein, the BRCA1 isn't hugely reduced, but it's more its ability to form the foci, which I think then, because that can't form the foci, then you do get the decrease in the RAD51. So that being downstream of it is more pronounced.
Yeah, so I guess I'm just saying maybe it's just something to bear in mind in terms of a biomarker translational, because the experience with PARP inhibitors is that it's not going to be useful for everybody and everything. So the key thing is that biomarker strategy. I'm not convinced. I don't think MIH2X is what you want to be looking at for a biomarker strategy. It would be RAD51. That would be my if I were to hedge my bets, primarily because of it's a good BRCA-ness biomarker.
Generally, a question that I had was, Emily's come up with a possible explanation of why we have indolent versus progressive EHE here. Do we have any other models for why that might occur? I know I discussed this with Brian a long time ago, and he didn't know what was causing that difference. But are there other models now that people have started to develop for why we have this indolent versus progressive EHE? So you don't have any challenges for your model, Emily?
I mean, I'm sure that like looking at senescence and stuff could easily be done both in patient samples and in some of the mouse models that have been developed already, I'm sure.
So Brian is not on the call. We have a paper that almost done, finished in there. So so this is exactly kind of what has been proposed. So it's a Cas-CAMTA1 expression from the endogenous locus coupled with CDKN2A knockout. So what we observe in this model, this is basically Caleb's work, is that, the mice die earlier. So it's kind of, you can say, aggressive. And another striking thing that we observe is that an increase in Ki67 staining in the EHE tumors. By the way, these tumors resemble EHE. I mean, they are histologically EHE-like. So just the knockout of the CDKN2A in a TC background is enough to cause, an aggressive phenotype. I mean, the aggressive in the sense, what by this mean is an increase in Ki67 staining, and then the mice, die earlier. So this fits with the clinically observed thing where, a secondary mutation in addition to the TC expression causes an aggressive phenotype. I mean, this is a mouse model, we all have limitations in there, like whether it correlates to a clinical phenotype, it's just we are one step closer to understanding what an aggressive EHE is.
Yeah, and I guess, AJ, what Emily is suggesting is a route that takes you into senescent cells and therefore indolent EHE. And Emily, what you've shown is that there's a knockout of CDK2, whatever it is, appears to make the disease much more aggressive. But I don't think anyone's saying that's the only reason the disease becomes more aggressive. There could be other mutations and other things going on in the cells that would make those cells more aggressive and bypass the arrest in the cell cycle, I guess.
Yeah.
The problem is, how much of genomic instability do we see in EHE? I mean, 50% of them have nothing. I mean, the same paper that you quoted, right? 50% of them is pure. There is no other secondary mutation other than Cas-CAMTA1. But if you say this is causing genetic mutation, genetic instability, but you would anticipate that there will be more mutations, right? But you don't see them.
Yeah, but in this paper, they only sequence the coding region of the most common tumor suppressor.
Right.
So you can imagine that you have mutation in non-coding region, and you would never see that. And it would take to sequence a full genome of the tumor to really see if there are other mutations. So this study was limited in the sense that it only looked at the most common tumor suppressors and oncogenes.
Yeah, and I think the thing is as well that the senescent onset of senescence would also explain a lower mutational burden, because the cells need to, in order to cause genomic instability, the cells need to not only acquire the mutation, but they need to proliferate afterwards. But if they're not proliferating, if they're just they're alive but they're not growing, you're not going to get a really genomically unstable tumor with loads of like secondary mutations.
Yeah, and this is quite common to most sarcomas. Like they have fusion protein, and there are very few secondary tumor burden, like mutation burden. I mean,
yeah, I don't think there are enough studies, at least whole genome studies, to to maybe in EHE, but other fusion-driven sarcomas, I'm not sure. One question, I don't know enough about these models, but I guess in your so-called senescence model, how is it possible to either sort it or grow it out to see if it naturally, if there are clones that become naturally aggressive?
mm-hmm.
So would it not, if you just keep them growing long enough, would they naturally accumulate whatever it is? I don't know what it is, but accumulate something that will basically.
If you leave them growing long enough, you start to get clumps, like little colonies forming in the culture. They seem to lose their contact inhibition. Obviously from that we can hypothesize that that's due to a secondary mutation, but without having sequenced it, we obviously can't say for sure.
Or if you could generate clones and then actually, look, so you start with the clonal populations and see which ones actually grow out and naturally acquire those mutations. I'm saying mutations, it could be epigenetic, whatever.
Yeah, of course, yeah.
But yeah, I think
Something's happening
progression, natural progression, if you will, which might give you some sense of. And I guess the other thing that's not clear to me, because I'm just looking at your measurements as well, how homogeneous or heterogeneous are these cultures? I mean, morphology or have you, I mean, because like you see, look at the measurements here, there's some variation, but I'm just wondering how homogeneous is it down the plate when you actually look?
Yeah, so you do get, in the DOX treated conditions, some of the cells don't ever express TASCAMT1. One thing we have noticed in the cultures, they don't really look any different just down like a normal light microscope, but I think it's quite pronounced in another picture, like here. The TASCAMT1 expressing cells, the nuclei tend to be really big and flat compared to the other ones. So you can see here you've got like a expressing cell and a non-expressing cell next to it, and it's a lot smaller. Not really sure exactly why that happens, but the peroxide treated ones seem to have a similar effect. So I think it could be something to do with the damage and maybe the early onset of senescence, because senescent cells do have this really broad flat morphology as well.
Well, sorry, I see Sorrow has been had a long time, so I'm not talking.
I'd almost forgotten myself, Paul. Thank you so much, Emily. That was some really nice data in there. It's lovely to get to hear about your work. I just wanted to kind of take a step back and ask what you think your data could mean for people affected by EHE.
So I guess the big one really with having found the impaired homologous recombination is that potentially PARP inhibitors could be useful. So I think this is really the next step now. What I mean, I don't have that long left, but what we're trying to do is kind of look a bit more at what inhibitors work within this. So again, looking at the PARP inhibitors and looking at ways that the DNA damage response can be inhibited to create some kind of like synthetic lethal response, because obviously these kind of inhibitors, a lot of them are readily available, like already available and approved and used in other cancers. So it's really kind of just looking at these now in the model and seeing if any of them could have a potentially clinical benefit.
And would you be up for writing a guest blog for Sarcoma UK about your PhD?
Yeah, I can do.
Yeah, I mean, one of the things I like about Emily's sort of thinking about PARP inhibitors, etc., is I love optionality in terms of looking at new therapeutic ideas. And because we can never guarantee that any particular route of research is going to actually work. And if it does work, we don't know how long it's going to take to develop into an idea. And we know that patients with HE vary significantly in their response to drugs. So there's something going on with HE that probably means if we come up with something new, it may not work for everyone. And so I really like the idea of having almost a stable of different ideas that are being researched as heading towards new therapeutic targets. Just so we're just maximizing the probability of producing something that's going to have a benefit. And at the end of the day, I just always remind people, our target is to stop people dying from EHE. That is our target. So any research that has a realistic chance of leading to some really interesting ideas is something which I just don't think we can afford to turn down. So I just love the idea. I mean, we've just been through a grant cycle process, or we're going through one, or I say we, the HE Foundation are doing that. And in that, there are three or four proposals, all of which are targeting a new therapeutic idea, but they're all looking at a different idea. And that, I think, is really fantastic. It's quite exciting, rather than having three that all want to do exactly the same thing, which is not nearly so good. Okay, well, Emily, listen, I know everyone's time is really tight, so that is fantastic. And as I said at one point, what I don't want to do is shut this down and then nothing ever happens from it. So what I may do is come around with a general email asking for people's thoughts, ideas. I don't want people to write pages and pages, just bullet points, ideas, things they think that based on Emily's work, things you'd like her to, or ideally we'd like to understand better. If you think there's something in Emily's theory that doesn't fit what you're doing or your understanding, what could we do to try and work out why that difference occurs?
Hugh, there are two more hands up actually.
Oh, I'm sorry. I can't see everyone. For some reason, I've only got 6 people. So yeah, but sorry, who's got questions? I'm just thinking people are going to leap in.
No worries. Just a question from my side. My name is Sandro Pasquali. I work in Milano with Nadia and Silvia. Nadia is at home with COVID, so she was sorry not to participate. So thank you for the invitation. It was a lovely discussion. I read Emily's work beforehand, and I really appreciate her presentation. Just a comment from my side, really. As you know, we have a PDX model of a primary EHE, which was a pretty aggressive tumor in the patient we sample, and we were able to generate this model. So I think that this preclinical model could be a good way to test hypotheses that Emily and team brilliantly generated. Like the possible effectiveness of PARP inhibitors, and it might represent also an opportunity to assess comparatively the possible effect of these drugs with drugs we are currently using. So I guess the information is very interesting, and there might be a way forward testing it in models that are closer to clinic to the patient work. Emily, any thought on this? Any ideas?
Yeah, I think that does sound really interesting. It would definitely be of interest to test these ideas in vivo. I guess that kind of also brings up another benefit of the model, really, is that we can use this to relatively quickly screen out some inhibitors that could be potentially useful, which can then be maybe used in vivo, looking at what's benefited in this model, see if then they work in vivo. It kind of saves a bit of time and everything like that, because we can quickly screen things now. So yeah, that definitely would be interesting to see.
I guess as a general follow-up comment, I guess to Emily and Valerie is, I mean, we ask a lot, you know, you present nice things. It's also to ask you guys what will you guys need from us, right? To you know, get a paper through, you know, finalize some validation experiments. If you need tissue samples, Sandro has offered his PDX models. So feel free to let us know what you need, and even if it's patient samples, we'll see whether we can make it happen from the ethics point of view. So just drop us a note. Yeah,
that would be brilliant. Thank you.
Yeah, that would be really useful to be able to access some patient sample to test some of the hypotheses of senescence and other. Thanks.
Yeah, definitely.
Yeah, sure. We can. Paul explain exactly what I thought better that I am able to do. So we are totally available to work together and to improve what we know and what we can do for patients. Really, I agree.
Paul, can you see anyone else with their hand up that I can't see?
I think
So yeah, I had one more comment.
Yeah, go on, John.
It's great, Emily. I think one thing that you didn't undersell it, but maybe other people, you know, it's not necessarily being captured as much, is that for a while, all of us working with this TAS-CAMT1 construct have had a hard time figuring out why we can't get cells to express this potent oncogene when we can get them to express any other potent oncogene we want. And so this DNA damage and the necessity of the CDKN2A or some other mutation, if that's the case, I think is important because it kind of helps us understand why it's so hard for us to get these cells to grow out. Because we've seen the same thing you have in any cell line we've put it into, and I know Brian's lab has as well. And it's even hard to get the mouse cells to grow out of the tumor without it. So I think it's a good explanation, and you should, you know, definitely sell that up a little bit when you go to write this up in the paper, that understanding why this oncogene it behaves differently than some other ones, at least in that phenotype. And you didn't express just like wild type TAS in your system, did you?
No, it's not something that that I've done.
Yeah, good work. Thank you for presenting to us.
Okay, guys. Well, if no one else has any other points or questions, it just leaves it for me to say a huge thank you to Emily for being the guinea pig in the first one of these sort of presentations. It's certainly been very useful for me. I hope it's been useful for others. And as I say, I will follow up, because really the whole point of this is just the starting point of a process where we want to try and pool ideas. And that's one of the key parts of this, is to try and harness. I mean, all of you have, as far as I'm concerned, the brains the size of planets. So, but if we can get all those planets working together and pooling ideas and theories and things, that energy and creative thinking can open up some really interesting ideas, I think. So anyway, thank you very much, everyone, for your participation. Really brilliant. Apologies for the glitch at the beginning. I'm glad we got over it. Thank you all for your interest and your support for EHE, because without you guys, the reality is we would be sitting here today with a group of patients who still had no idea or really hope of anything ever happening. And that's not the case. And that's thanks to you guys. So thank you very much. It really is hugely appreciated. All right.
Thank you, Hugh. Thank you.
See you all, guys. Okay. Bye-bye.
Bye-bye.